In this lecture, we're going to talk about signaling in the body, and then we're going to look at a plethora of ideas related to body systems that I think might be mentioned on the AP exam. And so I just want to expose them to you in case you see them on a test and they're not going to be so confusing to you. Let's talk a little bit about the endocrine system. When I'm talking about the endocrine system, I'm talking about glands and tissues that are secreting hormones that are traveling around the body and targeting other parts of the body, causing a response in other parts of the body. Now, we've talked about signaling over short distances. For example, we talked about how white blood cells can release chemicals and it can attract other white blood cells. For example, T helper cells. They can release chemicals and that can attract B cells and cause them to proliferate. Notice, this is signaling over short distances, or sometimes we call it local signaling. So our signaling molecules do not have to travel over a long distance before they reach their target. We just talked about transmitting of signals between neural cells, from neuron to neuron, or from neuron to a muscle cell. Those signaling molecules, those neurotransmitters, they have to cross the synapse. Again, this is signaling over short distances. When we're talking about the endocrine system, we're talking about signaling molecules, ligands, hormones traveling over long distances. Here we can see a cell is releasing a signaling molecule, and that signaling molecule is going to go into the bloodstream, and it's going to travel to a completely different area of the body, and we're going to have a response there by some target tissue in another part of the body. We've already talked about signaling molecules, ligands, and we've already talked about the difference between hormones that are lipid-based versus protein-based. So again, just to emphasize that your lipid hormones are going to be hydrophobic, they're nonpolar, and they can cross the membrane, and they're going to bind to a receptor, but that receptor is in the cytoplasm. Versus your protein hormones, proteins are large molecules, they cannot cross the membrane. There's usually charges associated with those protein molecules, and so they have to bind to receptor protein, and they oftentimes activate a secondary messenger. And the secondary messenger is going to activate other molecules in a cell. Lots of times they're kinases, and kinases will activate other molecules, and we have the transduction, or the spreading of the signal throughout the cell. I want to highlight this idea about proteins, that whenever this signaling molecule binds to them, they have what's called a conformational shape change. The reason why this protein is activating this protein is because it has a shape change. And that's how most proteins work. When a molecule binds to them, there's a change in shape, and that allows them to activate or react with another molecule. Once again, we've talked about signaling transduction pathways. There's three parts. We have reception of the signal. We have transduction, which is spreading of that signal from molecule to molecule. So here's our transduction. You can see that one molecule is activating another molecule. And then finally, we have a response. There's different responses. We can get genes activated or expressed, so this cell's now going to make a protein. We can activate organelles. We can activate specific enzymes. So there's many different responses. Way back in September, we talked about G protein-linked receptors. And then I have this box here because this box contains text that is from College Board, and it says, this is what students have to know. And if they understand something like G protein-linked receptors, then they should be able to understand and answer problems about signaling molecules binding to receptor proteins and causing a change in that cell, or initiating the signal transduction pathway. To emphasize this concept, I chose to teach you about G protein-linked receptors, and I'm just going to take a second to review that again. We have a ligand binding to this G protein linked, it's linked to a receptor, and that's going to activate, ultimately, this molecule called adenylyl cyclase. And this is the molecule that activates our secondary messenger. And I would know a secondary messenger. We talked about cyclic AMP. And cyclic AMP is basically an ATP molecule that has had 2 phosphates removed. So it's called cyclic AMP for monophosphate, mono meaning 1. And the secondary messenger is going to activate other molecules that will activate other molecules, and eventually we get a response in that cell. Cyclic AMP can activate molecules like kinases, which will activate other kinases by phosphorylating them. So remember that this would be a kinase and it's inactive, but if we add a phosphate group from ATP, we take off one and we turn it into ADP, and then we add that third phosphate to that kinase, it is now active. The fact that one molecule can activate many leads to the amplification of the signal, or the growing of the signal. The example that we talked about in class when we were talking about G protein-linked receptors is that you have cells, and we're going to talk about cells here in a second, that are storing glucose. So glucose is hooked together and it's stored in these cells. So I'm going to put G there for glucose. And the entire molecule is glycogen. We store excess glucose as glycogen. And ultimately, what happens in this signal transduction pathway is that the response is the activation of an enzyme. And this enzyme, now that's activated, can go and it can break up the glycogen, releasing the glucose molecules. So these glucose molecules then can be utilized for energy. So we're going to take the glycogen and we're going to break it up into monomers of glucose. One other point that I want to make is, remember we have an active kinase here because it was phosphorylated, and it's going to continue to be active unless we dephosphorylate it. And so kinases are deactivated by this process called dephosphorylation. That simply means that we're removing this phosphate. So our kinase is now inactive. Now, there's many organs that are part of what's called the endocrine system. And again, endocrine system just refers to parts, organs, glands that are releasing hormones. And these hormones can then travel and target other organs or other areas of the body. So the pancreas, the liver, your ovaries, the adrenal glands, parathyroid, all these are parts of the body that can release hormones and can target other parts of the body. Now, we do not have the time to go through and learn about all the different organs that we see here that are part of the endocrine system. So I'm just going to teach you about a couple organs related to the endocrine system so you understand how they can communicate with one another and how they're regulated through negative feedback. If I were you, I would make sure that by test day I have memorized how 2 different organs of the endocrine system can communicate and can respond to maintain homeostasis. The example I'm going to go through is going to target the liver and what occurs there, and the pancreas. These are involved in releasing and responding to insulin and regulating glucose concentrations in the blood. And since you already know a little bit about insulin and diabetes and blood sugar, then I think this is often the easiest one for students to read through and memorize. So let's look at what happens whenever you eat a sugary meal and you digest those carbohydrates and they're absorbed into your bloodstream, and as a result, your bloodstream now has high glucose levels. We have glucose levels that are higher than the target set point. You need a certain amount of glucose in your blood. You don't want too much. You do not want too little. So we have a target set point that we're always trying to maintain in our bodies. This excess glucose is detected by receptors. Since they're detecting chemicals, they're called chemo receptors. They're able to detect glucose in the blood. And these chemo receptors are found on cells of the pancreas. So this is the first organ that we're going to talk about here of the endocrine system. So the pancreas detects when your blood sugar levels are too high, And this causes the pancreas to respond. It's going to release insulin. We know that insulin is a protein hormone because it ends in IN. And this insulin is going to travel in the bloodstream. Remember, we're trying to communicate over long distances. And insulin is going to be received by muscle cells. It can also be received by liver cells. And I'm going to add and fat cells as well. This insulin is going to bind to receptors on these different types of cells. And it's going to cause these cells, the liver cells, the muscle cells, the fat cells, to take that sugar out of the blood. So to absorb it from the bloodstream. Now let's talk about how they get the glucose out of the bloodstream and how come they're not taking it out all the time. How come it's just whenever insulin is bound to the receptor proteins? Well, the fact that insulin binds to these receptors on these types of cells causes these cells to place, they're called Glute Transporters, and it basically stands for glucose. And we're talking about transport proteins. It causes these cells to place these transport proteins in the membrane. I'm going to show you that down here. Here we have the transport proteins. If you notice, they're not embedded in the membrane of this muscle cell, or this could be a fat cell. Whenever insulin, though, binds to this receptor protein, and you can see that here, then these transport proteins are now embedded in the membrane of these muscle cells and fat cells and liver cells. And now the glucose can travel through those transport proteins and into that type of cell for storage. So that's what I mean by insulin causes these glute transporters to be inserted in the membrane of those cells. The reason why these cells are taking up that glucose is so that they can store it for further use. So in 4 hours when you haven't eaten, then you have glucose supplies that you can use that would increase your glucose levels in your blood when needed. So this excess sugar is converted to glycogen. So it's strung together into glycogen and it's stored in those types of cells. Now, the result of all this happening is now there's less sugar in the blood. So sugar levels drop. And that stimulus, which is high sugar levels, is gone. So the end result basically shuts off this initial stimulus to this process. So this is an example of a negative feedback loop that is allowing your body to keep glucose levels at a target set point. Now we're going to take a look at what happens whenever you have low blood sugar. Once again, it's the pancreas that can detect that. And instead of releasing insulin like it did whenever we had high blood sugar, it's going to release a protein hormone called glucagon. Glucagon is going to travel in the bloodstream to those target cells, liver cells, muscle cells, and fat cells that were originally targeted to store glucose as glycogen. You can see here in this liver, here's a glycogen chain. We can break it up into glucose subunits that can be released into the bloodstream, and we can get that increase in glucose concentration in the bloodstream. Remember that these cells have these G protein-linked receptors, and whenever these signaling molecules bind to that, then it's going to cause this signal transduction pathway to initiate. So eventually we get enzymes activated that can break up that glycogen. We just talked about that a couple pages ago. Again, the binding to this receptor ultimately triggers the breakdown of glycogen into glucose, so that glucose can be released into the bloodstream, and we can get an increase in our blood sugar levels. Since this original stimulus is no longer present, the original stimulus was low glucose levels in the blood, then our end result eliminates that stimulus, and this is another example of negative feedback. On this previous page, there was a diagram that I didn't mention. I was going to go through both what happens whenever you have high blood sugar and low blood sugar. And this diagram shows these negative feedback loops. Again, this is showing you that the pancreas is detecting these original signals. When blood sugar is low, the pancreas detects that, and it produces glucagon that targets cells like those liver cells. That causes the breakdown of glycogen, and that raises blood sugar levels. If blood sugar levels become too high, then that again is detected by the pancreas. It releases the insulin. Then we're going to get storage of glucose from the blood, storing it as glycogen, and that ultimately then lowers those blood sugar levels. So again, we're trying to keep glucose levels at a target set point, and we're going to maintain that through negative feedback loops. One concept to note is that disease and other problems in the body occur when signaling pathways are altered. A good example related to this negative feedback loop that I just showed you is diabetes. Down here we can see that cell signaling pathways are altered. They're not functioning like they should. And so as a result, disease sets in. There's two types of diabetes. We have type 1 diabetes. Now this is caused by an autoimmune response. For some reason, our own white blood cells will attack these pancreatic cells that are producing insulin, and they're destroyed. So the cells in the pancreas can no longer produce insulin to help regulate blood sugar. With type 2 diabetes, there's some kind of defect in the signaling process, and we're not able to take up glucose from the bloodstream. So individuals have to carefully maintain their blood glucose levels by watching their diet. These two diagrams are simply trying to show you what has happened, what are the changes to the signaling pathway related to both types of diabetes. We have diminished insulin because those cells in the pancreas were destroyed. They're called beta cells. And so we are not receiving this signal to insert those glute transporters so that they can actually take glucose into the cell for storage. Here we see insulin resistance. For some reason, we're not able to respond to insulin when we have type 2 diabetes. Insulin is being produced, but there's a defect in the signaling pathway and is preventing the uptake of glucose from the blood. So therefore, when you have diabetes, your blood sugar levels are much higher than normal. I wanted to show you some of these other concepts that you have to know that are indicated by College Board. You can see that there's many ideas that you have to understand, and I can use these different examples to explain them to you. And if you notice, all of these examples are related to what we just talked about with the pancreas and liver cells, the insulin and the glucagon. So let's take a look at these. You have to understand that there's interaction and coordination between organs. So we talked about the pancreas and the liver as an example. In multicellular organisms, it's those signal transduction pathways that coordinate activities so that we can have a response in the organism. So remember, whenever epinephrine and glucagon binds to those G protein-linked receptors, then it sets off these signaling transduction pathways. Endocrine signals are produced by cells, and they travel over long distances. Insulin is produced by the pancreas, and it's traveling over a long distance through the bloodstream. And then we talked about membrane proteins. They play a role in things like facilitated diffusion, moving those charged and polar molecules through a membrane. We had to insert those glute transporters so that glucose could then come into those cells. And then finally, we talked about disease sets in when we have alteration to these feedback loops. An example of that is with diabetes. One thing I want to note is that animals aren't the only organisms that can produce hormones. Plants can produce hormones as well. And these hormones can travel over long distances throughout the plant tissue from cell to cell. Remember, cells are connected by plasmodesmata. So let's indicate that. It's been a while since we talked about that. And molecules from one cell then can move to another cell through those gaps, those plasmodesmata. This diagram was on one of your worksheets, so I want to take a second to explain it. Now, even though I don't have to specifically teach you about any plant hormones, sometimes it helps if we go over some plant hormones because you might have questions about them. This is a diagram that was released from the 2015 test, and it's about a protein hormone called auxin that's produced by plant cells. Auxin specifically is produced here. It's produced in the tip of the shoot, and auxin migrates to the dark side of the stem. Light is the stimulus that causes these cells to produce and release auxin. Let's take a look at what happens whenever we alter the tip of this plant. As long as the tip is intact, then this cell is going to receive that stimulus. Auxin is going to be produced. Let's show it migrating to this dark side of the stem, and that's going to cause these cells to elongate and divide so we get more growth on this side of the plant. We've briefly talked about this in a different lecture, and I showed you a diagram about how receptors are going to bind to auxin, and it's going to cause signaling transduction pathways that can result in the elongation, division of these cells on the dark side. And if we have more growth over here compared to here, then that plant is going to bend towards the light. So that's why the light is being shown coming from this direction. And the plant is responding and is bending so that it can grow towards that light source. Since the tip produces auxin, if we cut off the tip, no auxin, no bending. Because it's the auxin that is actually causing this response in the plant. For the shoot that you see here, it says the tip is covered by an opaque cap. That means light can't penetrate. And you can see there's no response. So that's telling you that light is the stimulus. So there has to be photoreceptors in the tip that can receive that stimulus, then causing the production and release of auxin. Here we've cut off the tip. We've put a membrane here, but that membrane is permeable. And then we replace the tip back onto this membrane. We have the tip present. We have the stimulus. We have light that can be received by these cells. So these cells can produce auxin, and since this barrier is permeable, then the auxin can travel through that barrier, and then it can cause those cells to elongate. This is showing you that we cut off the tip and we separated it by an impermeable barrier. These cells are going to produce auxin, but they cannot travel through that impermeable barrier. So then these cells aren't receiving that stimulus to grow. So this is an example where you have to be able to look at results and then make decisions, draw conclusions about what is taking place. So here's an example of a problem where we're dealing with a plant hormone. Another plant hormone that you might be familiar with is ethylene. And the reason why I'm teaching about ethylene is because ethylene is related to a positive feedback loop in plants. So far we've only discussed negative feedback loops, which allows us to keep a set point or a certain condition in our body. Now we're going to take a look at a positive feedback loop, and I'm going to give you two examples. So one is with the ripening of fruit, and another one is with childbirth. We're going to talk about that on the next page. Whenever cells ripen, whenever fruit ripens, what is happening is these cells are actually breaking down. The sugars are breaking down, their cell membranes are breaking down, and that's what's happening whenever a piece of fruit is slowly ripening over time. Now, whenever these cells break down or atrophy, same thing, then these cells are going to release ethylene gas. Ethylene gas actually triggers more ripening and more breaking down of these cells. So ethylene gas itself increases the rate of ripening. More cells are going to start to break down because they're receiving the signal, the stimulus, which is ethylene gas. So these cells are going to break down. Remember, breakdown produces even more ethylene, which is going to cause even more cells. Ultimately, we're going to end up with a final event, a culmination. So the culmination of this event is this fruit is ripe and it falls from the tree. So when we're talking about a positive feedback loop, there's always an end result. And that stimulus is removed, so all the cells are broken down, so no more cells are producing ethylene, and we end up with this final event. Childbirth is another easy example for you to remember that is showing us positive feedback. The official definition of a positive feedback loop is whenever we have a condition and it's intensifying, and the result actually causes the condition to intensify further until there's a culminating event. I'm going to start here on this diagram. So after 9 months and that baby's grown and it's gotten heavier, then it starts to put pressure on the cervix. So here's the cervix and the head is putting pressure on the cervix. This sends a signal to the brain. So I'm just going to show a signal going to the brain. And the brain, receiving that stimuli, is going to secrete another hormone, which is called oxytocin. Oxytocin then causes these muscles to contract. If you think about that, if these muscles are contracting, then that's going to cause the head of the baby to put more pressure on the cervix. That's a stimulus that sends a message to the brain, and the brain is going to cause the release of even more oxytocin. So contractions intensify. which again causes the head to put even more pressure on the cervix. So you see what I mean by we have a condition that intensifies, and the result is that head is pushing on the cervix, causes this condition to intensify further, because this pressure again causes the release of more oxytocin, which causes more contractions, which again causes an increase in that pressure on the cervix, until there's a culminating event, and in this case, it's childbirth, and that stimulus is completely removed. College Board says you need to know an example of a positive feedback loop or a positive feedback mechanism, and a good example is childbirth, or you can even talk about fruit ripening. Remember that reproduction is only going to occur when we have an excess of free energy. So if we have insufficient free energy, this results in loss of reproduction. If we continue to have loss of free energy, then we're going to lose the ability to grow, and then eventually it can lead to death. Before I get into some ideas about development, I just want to take a second to explain why certain body systems and processes have been earmarked by College Board for AP students to know. There are many diseases that we have yet to cure. So we're trying to give you information most related to some of these diseases, so that maybe someday you can go on and you can do research and come up with ways to treat these diseases. So we first talked about the nervous system. We cannot cure paralysis, Alzheimer's, Parkinson's. That has to do with the disruption of the functioning of neurons. So if you understand neurons and how cells communicate, then we might be able to come up with some treatments for those types of diseases. Then I went into the immune system. Understanding how we respond with our immune cells is critical for us developing new treatments that can kill bacteria and viruses, or even prevent us from becoming infected by certain viruses. Think of all the research that's being done now because of this new virus, this Zika virus, and we believe it's causing microencephalization. It's carried by mosquitoes, and it's spreading to many parts of the world. Understanding how viruses function and how we respond to them is critical for us developing vaccinations and other treatments for such a disease. When we have disruptions in cell signaling, that's when disease sets in. We cannot cure diabetes, and we do not understand why people develop type 2 diabetes. So you have to understand how cells receive signals and respond to signals, so that we can then go in and maybe fix those signaling pathways, and we can reverse this disease. And then finally, we're going to talk a little bit about development. With development, we do not know a lot about human development simply because there's been restrictions of testing on human embryos. Only recently has a researcher gained approval in a different country to take human embryos and alter these genes that guide development, so that we get a better understanding of how these genes relate to human development. And is there something that we can do to alter these genes, fix these genes early in development, so we can make sure that this organism, this child, develops correctly? That's why we're going to take a second to go through some ideas related to development. Timing and coordination of events is key to development. We get the formation of a zygote, and the zygote starts to divide by mitosis, and eventually we have undifferentiated cells of this embryo that have to differentiate. Differentiation of cells is due to 4 key factors. So the first factor that we're going to look at are these developmental genes that I just mentioned. They're called Hox genes. We've gone into other organisms, into embryos, and we've mutated Hox genes, and we can see then what mutations in these genes do to the development of the organism. Differentiation of cells is also affected by unequal distribution of cytoplasm whenever those cells divide. We're going to talk about induction and how it influences cells to differentiate and develop. And then we've already mentioned apoptosis is critical to normal development. First, we're going to look at genes that guide development. Now, these genes that guide development have a fancy name. They're called homeotic genes. Many times they're referred to as Hox genes. Hox genes guide where body segments are going to develop, where appendages are going to develop, so they have a major role in the development of organisms. Mutations in these Hox genes lead to major developmental abnormalities. The fact that we have Hox genes in common with many other species indicates a common ancestor. So sometimes you'll have questions about evolution related to Hox genes. Just like all genes, Hox genes can be turned on and turned off, and it's critical that these genes are regulated, that they're expressed at certain times or they're turned off at other times. And these genes and their expression can be affected by things in the environment, like temperature. And we'll talk about how temperature actually affects which sex develops in reptiles. Now on this next page, I wanted to give you an example about how the environment can alter gene expression. In this situation, we have a change in the environment, temperature is changing, and it's going to affect whether eggs will develop into female reptiles or male reptiles. Temperature affects sex determination of some reptiles. Make sure we can read diagrams like this. When we're looking at this species of turtle, if these eggs are incubated at a temperature between 26 and about 28, then all 100% of the eggs are going to hatch out and they're going to be males. If you take these exact same eggs and you incubate them at a temperature closer to about 30 degrees Celsius, then this is saying that only about 25% of those eggs are going to hatch out to be males, and about 75% of them will hatch out to be females. So temperature is affecting whether certain genes are expressed or not. It's affecting gene expression, and it can cause changes in development. So again, this is just an example to show you that environmental factors can affect the development of organisms. Now I want to take a second to review how gene expression can be altered or affected. We've already talked about this in the molecular genetics unit. Remember that junk DNA, so I'm going to draw some DNA here, and we have areas that are considered genes, but then we have these other areas that are considered non-coding regions. But we have since learned that they code for microRNAs. So these microRNAs are produced, and they will then form these hairpin structures when they fold upon themselves, and you can see that here. These microRNAs, these RNAi molecules, I stands for interference, they can be processed by dicer, and here you see dicer is going to process this molecule and basically cut it up into smaller fragments that are considered these microRNAs. These microRNAs also bind to some proteins. They were called those Risc proteins. So let's show that there's some Risc proteins bound to this microRNA. And it forms a complex, and these complexes can do different things. So we can alter gene expression, whether the gene that coded for this mRNA is actually going to lead to the production of a protein, or is it going to be silenced. When these microRNAs bind to messenger RNA, it can do a couple different things. It can cause the destruction of those messenger RNAs, so they're not able to be translated, and you can see that right here. Or it can block the ribosome. Here we have a microRNA Risc complex, and it's bound here to this different mRNA. And when ribosomes are attempting to translate this mRNA to build a protein, they're not able to complete that protein. So we say that the gene is not being expressed, or the gene has been shut off. Also, remember that alternate gene splicing can affect which proteins are going to be made in a cell. So it's affecting gene expression. Here we have a gene. Remember the introns, are cut out, and the exons are spliced together. So here's our exons. Exons will exit the nucleus, so the exons are spliced together, and we have that processed mRNA that can leave the nucleus. If the exons are spliced together in this arrangement, you get this protein. But if they're spliced together in a different arrangement, then you get a different protein, all from that same gene. So again, we can have different proteins being built in the cell. So we're getting a change in gene expression, and this can happen due to different signals in the environment, or different cells expressing or making different proteins according to the same gene. Okay, so the second factor we're going to look at is just simply the uneven distribution of cytoplasm during mitosis. As a zygote divides into two cells and those cells divide, That division with no increase in size is called cleavage. So when this cell splits in two, then the entire structure doesn't get bigger, it just cleaves into two cells. So that's what we mean by cleavage. So as these cells cleave or divide in two, you can see that you get an uneven distribution of molecules. And so there's different transcription factors, there's different proteins and enzymes that get distributed into these cells, and it's going to affect the genes of those cells. Different genes in that nucleus are going to be turned on compared to that nucleus. Those same genes are going to be turned off, and it's simply due to the chemicals that were distributed into the cytoplasm. Next, we're going to look at how cell communication from cell to cell can actually affect the development of the cell. And cells can communicate. They can communicate through chemical signaling and through physical contact. Now, any chemical signal or physical signal that causes a cell to differentiate or develop is called a morphogen. So it's a chemical signal, like a morphogen, will cause that cell to morph into a certain type of cell or differentiate. We can see chemical signaling happen in this situation. So here's the membrane of one cell. Here's a membrane of a nearby cell. So this is signaling over short distances. And this cell is releasing chemicals that's going to bind to that receptor protein, and it's going to activate certain genes in the nucleus. If the cell didn't receive those chemicals, then it would not differentiate into a specific type of cell. Only after it receives the stimulus is it going to do that. So here's an example of how like direct contact or physical contact can activate a cell to express certain genes. Here we have a protein, and by directly binding to another protein of another nearby cell, then we're going to cause gene expression to occur and this cell to develop. On this third diagram, we're seeing chemical signaling. It's just showing you that chemicals don't have to cross a synapse and bind to a receptor protein, but there's actually gap junctions. And remember, in plants, we call those plasmodesmata. And chemicals can be sent from one cell's cytoplasm to another cell's cytoplasm through these gap junctions. On this page, we're still kind of continuing with how cells can signal one another through chemical signaling, through physical contact. There's a little more detail than what you need to have memorized, but I just want you to be familiar with, we're talking about development of embryos. Whenever this embryo is just a few cells big, it kind of looks like a hollow basketball, and you can see that from here to here, that this hollow circle kind of folds up and back into itself, and this is what we mean by gastrulation. But what I'm trying to point out is in early development, we get the formation of these three main types of tissues: the mesoderm, the ectoderm, and the endoderm. And this is what we mean by germ layers. And the cells of these germ layers are in contact with one another. They're releasing chemicals that affect one another, and that's going to cause these cells in these germ layers to actually differentiate into different tissues of the body. The endoderm, which is, again, it's just a layer of cells. These cells are signaled to turn into like cells of the digestive system. The ectoderm, these cells are receiving signals to turn into kind of external tissue like skin and nervous tissue. And then we have the mesodermal tissue, or the mesoderm. These cells, you can just cross through that, these cells are differentiating and turning into like cells of the heart. You don't have to have that memorized, but you just need to understand that here we can see in this diagram, there's different cells of the endoderm, the ectoderm, and the mesoderm, and they are signaling one another. And that is the reason why these cells will differentiate and turn into heart cells. If these cells were in a different area, they wouldn't be receiving those signals. They wouldn't differentiate into these types of tissues that are necessary for development. Now, there is kind of a famous experiment that shows that cells produce specific signals that cause other cells to develop. This kind of relates to this structure that I showed you up there. I don't think you'll be able to tell where exactly we're at, but in early development, there is a structure that forms right there, and it's called the notochord. The notochord basically is going to turn into your vertebrae in most organisms. Now, what has been done in experiments is this notochord has been cut out and it's been moved. So let's cut it out and let's move it over here. Then what we see, no matter where we put that notochord, the cells in the area directly above it will differentiate and turn into cells that form this neurotube, which basically house your spinal columns. No matter where we put this notochord, it's going to release signals, and the cells directly behind it are going to differentiate into cells of the neurotubes. This is just an example that cells are communicating, and that is critical to development. And then apoptosis is basically programmed cell death. And we think usually of cell death as something that is bad, but it's a normal part of development. So a couple examples of where apoptosis is necessary is in the defining of digits. And you probably even know some people that were born with like 2 toes attached together, even 2 fingers attached together. There was a disruption in cell signaling, and apoptosis didn't occur in those cells. Tadpole tails, they don't just fall off. What happens is these cells die by apoptosis, and then all of the molecules and nutrients that are tied up in those cells get absorbed by that developing tadpole. We're not losing nutrients by just getting rid of an entire structure made of cells. Okay, so finishing up just a few odds and ends. I think the Bor Shift is pretty complicated, and I've seen lots of questions about the Bor Shift, and then we're also going to talk about the countercurrent exchange, and then be done with this unit. When we're talking about the Bor Shift, we are talking about something related to hemoglobin. And remember, hemoglobin is a protein that is in red blood cells, and hemoglobin binds to oxygen, or we say it has an affinity for oxygen. Whenever you are exercising, you're going to be using ATP, and you have to make more. And in the process of respiration and in making of ATP, you also produce CO2. That CO2 is going to leave that tissue, go into the blood, and your blood contains water, and now it has a large amount of CO2, and they react together and they make carbonic acid. Because of that acid, the blood pH drops, so it becomes more acidic. When blood becomes more acidic, the hemoglobin in those red blood cells has a lower affinity for oxygen. It doesn't want to hold on to it. It actually has more of a tendency to release it. So think about the benefit for that. In an area where there's lots of CO2, those cells are using a bunch of ATP. They need to make more, and so they need more oxygen to do that. Basically, the acidity in the environment allows hemoglobin to release more oxygen than what it would have released whenever there wasn't such a demand in that area. This graph is showing you the Bor Shift, and it's simply showing you that whenever blood acidity or pH drops and it becomes more acidic, then this much oxygen is getting released. And if the blood wasn't acidic, then that oxygen would still be bound to those red blood cells and not being released by the hemoglobin and being delivered to the cells of the tissue. And then finally, we're going to look at countercurrent exchange. In countercurrent exchange, basically there's kind of 3 big ideas. One is you have 2 flowing bodies, and it's important that you understand they're flowing in opposite directions from one another. And these flowing bodies are also going to be in really close proximity to one another. So we're going to talk about how countercurrent exchange helps organisms that live in cold environments regulate their body temperature. So picture this animal walking on ice and snow. What happens in the situation is blood has to deliver oxygen to these tissues. So blood is leaving the body and it's really warm. As it flows down to the tissues and it's getting further away from the body, it's actually dropping in temperature. And it's dropping in temperature due to a couple different reasons. Whenever the blood drops off oxygen, it stays in the blood vessels, and then it's going to return back through a vein and then to the heart. Let's take a look at what's happening here. As this blood is returning back to the heart, notice that it's right next to this blood vessel, and the blood in this blood vessel is warmer. And just like molecules move from high concentration to low concentration, Heat moves from a high area of heat to low areas of heat. And so if these blood vessels are in close contact with one another, then heat from that warmer blood is going to move into that colder blood, and it's going to warm it up. It's warmed up now to 14 degrees, but that's still colder than this blood that's coming from the body. So more heat is transferred, and as you go up, you can see that this returning blood is always just a little bit colder than the blood that's leaving the body. And so heat is continually moving, and you can see that even though this blood got sent to an extremity, it's returning back to the body, and it's still very close to the temperature of the blood that was leaving the body. And this is important because if cold blood was returning back into the body, then the body temperature of this organism would drop, and they would need a lot of free energy to try to maintain body temperature. Lots of times that's not available to your upper level carnivores. Countercurrent exchange, in this situation, it's helping organisms maintain a constant body temperature. That heat's going to move from areas of high heat or blood that has more heat to the blood that has less heat. But it's warming the blood before it returns back to the body. And we also see countercurrent exchange when we look at the gills of fish. The gills of fish are loaded with blood vessels. These blood vessels are picking up oxygen from the water. We're going to have two flowing bodies, and they're going to be in opposite directions of one another. Like the lake water is going to be flowing in the opposite direction that the blood is flowing in the gills. We have water flowing this way, and we have blood going in the opposite direction. So you take a look at the relative concentrations of oxygen in the water and in the blood, that here, whenever the blood's just getting to the gills, it's low in oxygen because it needs to pick up oxygen. It's returning from the tissue, so it's low in oxygen. And it's lower than the oxygen in this water. And so oxygen moves from the areas of high concentration to low concentration, and so as the blood moves, it's picking up more and more oxygen. So again, we have an area where the water has higher concentration of oxygen than the blood that is flowing near it in a blood vessel, and so more oxygen is moved over. By the time the blood makes it through the gills, then it's picked up a lot of oxygen from the water, and it's going to then carry that oxygen to the tissues of the fish. And that includes our very last video.